So good morning, everyone. It's morning here, and I'm not a morning person. So personal request, if anyone is listening and engaged and you have questions, please feel free to stop me and ask questions. It would be more interesting for me if this is more of a dialogue than a monologue, even if it means I don't have time to say everything, it'd still be more interesting. So I'm going to talk about two projects. One is the collaboration many people here did together on the paper that should be accepted any day now. And the other one is a sneak peek on my next paper that I'm trying to present everywhere so I can get feedback on it before I submit, essentially. So this project, the systems biology analysis of lung fibrosis-related genes in the bleomycin mouse model is a mouthful, but I'll explain it a bit. This is a collaboration between Vadim's group and Robbie's group, and this project is spearheaded by Mitya, and I was fortunate enough to be allowed to collaborate. And the design is pretty straightforward, and this is a design we see a lot, right, in our groups. We take a phenotype, we ask what are the functionally important genes for this phenotype, and then we analyze the genes, right? It sounds very, very simple and straightforward. God is in the details. The phenotype we're talking about here is lung fibrosis. And lung fibrosis is, well, fibrosis in general is an age-related phenotype. It happens more with age, and it's thought to be part of the aging process. Although we're not sure about the relationship exactly between fibrosis and aging. And it does happen a lot in the lungs. In humans, we see a lot of what we call IPF, idiopathic pulmonary fibrosis, which is a disease we know almost nothing about. It's a lethal disease of age. And in this case, the functionality of genes was assessed as we did previously with longevity genes. If we asked if we manipulate a gene, is it going to affect the fibrosis outcome? And it can promote fibrosis, in which case we call it a pro-fibrotic gene, or reduce fibrosis, in which case we call it an anti-fibrotic gene. The problem is you cannot do these experiments on humans, obviously. So you have to do them on mice. The only model of lung fibrosis on mice, the best one really, the most used one, is called the bleomycin model, where they let mice breathe, inhale bleomycin, and that induces lung fibrosis. It's not exactly the same as the chronic fibrosis we see in all people, but it's similar. It has a lot of similarities. And this is the part where a lot of the legwork is. You got to read all the papers, you got to find all of the manipulations done and assess whether they really affect fibrosis or not. And that's the manual curation step. And that can also result in a database, which we have, of genes that affect fibrosis, lung fibrosis. And most of the paper does the analysis on this gene set, and I'm not going to show everything. It's quite long, but I would like to show what I think are the three most important and interesting findings. So first of all, as a side note, these are the manipulations generally used to assess the impact of a gene on fibrosis. And you can see here, most of the manipulations are either knockout or overexpression. And that makes sense. These are the easiest to perform and have been done for many, many years. And the important side note is we see a lot of consistency between the direction of the influence of the gene in the mouse bleomycin model to the expression of that gene in human IPF. And that's important because it means the bleomycin model has some relevance to human IPF. Not exactly how much, but it has some relevance at least. It's not completely irrelevant. So what I think are the most interesting findings, first of all, when we look at the evolutionary conservation, and this is the same assessment as Pedro showed before, where you look at the percentage of orthologs between human and other species. And what you see in green is the total proteome. What you see in the orange triangles is just the percent of orthologs from the fibrosis-related genes. And when Mitya did this analysis, he immediately saw that there's a very, very sharp cutoff between vertebrates and invertebrates, where in vertebrates, the fibrosis-related genes are over-conserved, and after that, they're under-conserved. So something in fibrosis seems to be vertebrate-specific. And we don't know exactly what. We can hypothesize. We think it has to do with adaptive immunity, which is more present in invertebrates, but we don't know for sure. But it's a very interesting finding, I think. The second one is the enrichment analysis. So I did this very big slide, but essentially we attacked this in 3 different ways. We did a network analysis, and you all know about the network analysis. And Mitya also did the human-based network enrichment-based analysis, which is kind of like doing a network analysis, finding subnetworks, and then seeing what are the subnetworks enriched for. And then we also did the classical and gene enrichment analysis. And the interesting thing we found, except for the fact that we found, interesting pathways that are enriched in fibrosis, the most interesting thing we found is that, first of all, both the pro-fibrotic list and the anti-fibrotic lists, both of them form networks, protein-protein interaction networks. And they show different enrichments between them, where in the pro-fibrotic list, we see, for example, a positive regulation of cell proliferation. In the anti-fibrotic, we see a negative regulation of cell proliferation, which makes biological sense. We also see a very strong immune component in the pro-fibrotic list, but not in the anti-fibrotic list. So the differences are very interesting, and I think bring something new to the table of people when people look into fibrosis and try and see what promotes fibrosis and what stops it. We see different things, different pathways. The third thing I wanted to show is, I think, the most interesting finding. We compared the list of fibrosis genes to the list of longevity-associated genes from GNAs that was presented before. And we see a very, very high correlation between genes that are pro-fibrotic would be anti-longevity and vice versa. That's very, very important because that indicates that, first of all, fibrosis and aging seem to go hand in hand. But the fact that these are manipulation-based lists, to me, indicates that this is a mechanistic, it's not just a correlation, it's a mechanistic link. And to me, it indicates that fibrosis could really be a mechanism of aging. It's a bit of a jump to say it as a fact, but that's what the indication is from these results. And so we put all of these results in the database, and the paper, I think, should be accepted probably this week, right? Hopefully. So this was this project. The next one I wanted to show is the next paper I'm planning to submit, and I asked Robbie to include it in this presentation because I'm trying to show it to as many people as I can, as I said before I submit. So let me see how much time do I have?
Yeah, I think it's okay. Don't worry about it.
About 10 minutes?
Even more. It's fine.
Okay. So it's a big project. I'm not going to show everything, but at least the basics. So the project is about aging red blood and the patterns we see in the aging red blood and what we think it shows us. And the main question when we present this is why even work on rats, right? Why even do research on rats? Rats kind of been in a decline as a research model since the 1980s when the mouse transgenic models started to be presented. So obviously everyone moved to mice. Mice are also cheaper. They're easier to house. You get more per space. They eat less. So everyone kind of moved to mouse, and rat really is in a decline as an experimental model. But it has a lot of benefits. The first benefit is the physiology of the rat is much closer to human than the mouse. still closer to a mouse than to a human, but it's closer to a human than a mouse. And the same goes for some of the blood parameters. One of the main differences in humans, we see an aging disease called AML, acute myeloid leukemia. It's a form of leukemia. When people talk about leukemia, usually they talk about child leukemia, but leukemia is an aging disease. And mice don't display any myeloid leukemia, none. They have a lot of lymphomas, but no leukemia, no myeloid leukemia. And rats do have an aging-associated myeloid leukemia. So in terms of hematopoiesis, which is what the lab I'm in now deals with, we're in a hematopoietic stem cell lab. The rat seems to be a more relevant model. The lifespan is more feasible. It is not more. It is feasible. It's not much more than the mouse. The NIH has an aging colony of rats, so I can essentially get rats of any age at any time. So they're available, and they should have a higher bone marrow cell number, more than the mice, so could have more cells to work with. So it's a relevant model for us. Now, a few months ago, my lab, in collaboration with Morgan Levine from Yale, she used to work with Horvath. She was his student. They generated a rat aging methylation clock. And we jumped on that data because what they did, one of our people, you see here, it's called Chris, as they were getting cells to the methylation analysis, he was analyzing the blood populations using these antibodies listed here. It's kind of a new panel, kind of a standard panel with some new antibodies. And the reason I was interested in this data is you can see here, these are how many rats we have of what age. And the fact that they were representative of every age, every month of age, got me interested. I thought this was interesting. So I started analyzing this data. Problem is we don't know a lot about rat blood and what markers the cells should express, what antibodies we should use to see exactly what blood population. So I used kind of semi-advanced methods to identify blood populations, just use TSNE and self-organizing maps to cluster different cells together and then look at what markers they express and then define some of the populations that we could define. We couldn't define all of them. We could assess their aging changes, and we saw some changes that were to be expected. People that deal with hematology know, for example, that B cells are expected to go down with age. Monocytes are expected to go up with age. Some expected changes. Some of the changes were unexpected, like an accumulation of a subset of T cells that we completely did not expect. But I'm not going to go into too many details here. The interesting thing we found, they tried clustering the rats by their blood parameters. And I thought I would be able to see some sort of prediction of disease. But I was surprised to see that when I clustered them, just simple hierarchical clustering, they clustered by age. And they clustered by 3 major age groups: young, middle-aged, and old. With some overlap between the age groups, but overall clustered by age. So it kind of surprised me, and I thought, well, if they clustered by age, can I predict age just by the frequency of blood populations, population frequencies of blood cell types? So I ran a very, very simple least square linear model just to predict age based on the population frequencies. And it actually got a pretty good result. I was able to predict age with an RMSE score with a range of 2 months, right? Plus minus 2 months, which was kind of surprising. And also the grayed out dots here are the animals that had a pathology, an overt pathology that we could see. And so I excluded them from the model when I made the least square model. And then obviously when I look at residual age, or how far they are, how far the rat expected age is from his real age, The more sick the animal is, the farther away it is from its expected age. And you can improve this. We don't have enough rats really to do this improvement here, but we did kind of a simple neural network, neural net, just to say we did machine learning basically. But we just wanted to see if a neural net can predict it even more accurately, and obviously it can. I think it's a bit over-trained model, but the point I think is important here. I think it can also be applicable to humans. Yes.
Have you any idea why CD25 and CD4 are so with such a high predictive power?
Because they change linearly with age in a very, very stable fashion between the rats. And this is completely new. We had no expectation of CD5 even being important, CD25. But it turns out it is. It's one of the new things we found that's surprising. We don't know why. But this is the T cell, the subset of T cell that gets accumulated with age, are CD4 T cells that also express CD25. And I'm not exactly sure what they do. The literature isn't also very, very clear on what they do.
Okay.
Well, actually, I see that you have some outliers. I mean, I don't know how large is the sample size?
It's about 160 rats.
So probably neural networks may not work well. I would suggest having some tree-based models to see if they can capture the outliers better. That can be an idea. Also, I had a question about previous paper. For a fibrosis paper, did you see any patterns with matrix metallo proteinases expressions?
I think not. Maybe Mitya can correct me, but I don't think we did.
I also don't remember seeing them.
We just do not look for expression patterns, you see, because we use or knockout or overexpression. So 0, 1, and that's all, without any expression values.
It's also important to note that we see in these analyses the very, very obvious things that just stand out. For example, there was no enrichment of cellular senescence. But when we look at the different pathways that are enriched, they do kind of converge or can converge on cellular senescence. So a lot of things we didn't see but are probably there. Okay.
So regarding the sources that you used, how many things did you look at? I mean, so, can it be more general than just CD25? Maybe many CD.
It could be. So we have these 7 antibodies, and then some general information that I didn't put here, but body weight and stuff like that we can also use. But generally speaking, we have these 7 antibodies, and then we have indexes or parameters that we can extrapolate from that. Right? So we don't have to just look at levels of expression of each antibody. We can do combinatorial expressions and subpopulations. And I looked at about, let's say, 15 parameters. So it's a very low parameter number. That's why I use the least square and not, machine learning, because this is really an overkill for.
The more features you will have, and with this limited number of data points, the harder will be for machine learning to.
But my point is
overfit.
My point is we don't need the fancy DNA methylation with 100,000 features training model to predict age. We can do this with this very simple population. You can just do a blood test with cell numbers, and you can probably predict age pretty accurately, and I would argue predict pathology also. That's my main point. Yeah.
One more. Are there any data regarding CD25 and CD4 in other rat strains, say Sprague-Dawley, or I know?
No. No. But maybe one strain, the brown Norway, but not a lot. There's not a lot of information on CD25 in general.
Okay.
One other idea.
Take the NACMOL rat from Vera Garbunova and test.
Good point.
Yeah, but testing that will you will need a large colony.
But probably there should be public data on CD25, CD4 that you can use to actually check other strains.
Yeah, I could extrapolate from gene expression. There's not a lot of data on CD25 surface marker, right? Because that requires flow and a good antibody, and people need to actually
what
run the flow.
about
But gene expression.
Actually, you can deconvolute probably for CD25, CD4.
You mean from gene expression?
Yeah, there are deconvolution tools. I played a bit with some of them. I mean, I cannot guarantee that we exactly will get CD25, CD4, but they give estimations for appropriate for different cell types. So if we take rats, blood, RNA-seq, then apply immune CCD, is that we apply the convolution, maybe we can actually get cell types, but maybe not. It's worth checking.
Yeah, no, it's a good point. I could definitely do it on the human data and see if we see anything similar.
That's what I wanted to say. Maybe it's a stupid question, but why not use human data? Because you are implying from this that CD25 is really important for aging, because otherwise it wouldn't predict age. So then it should be universal. It should, or at least for mammals, something. So for humans, you have the best chance probably to have this kind of data in large quantities.
No, because CD25 is normally not looked at.
It's not what? Sorry?
It's normally not looked at.
Okay.
So we included it because someone thought it might be interesting because they read a reference somewhere, but it's not a common antibody that's used to differentiate T-cell populations.
But at least if you start a new study,
yes,
from
definitely.
humans, it will be easy to get blood, the easiest.
The new studies that use advanced cytometry machines where you can use 16+ colors like Symphony and all that stuff. They might be using CD25, although I'm not sure. Usually people stop at CD4, CD8 T cells. That's it.
Yeah, those are the classical ones.
Could you remind for CD25 how predictive are they on mice? Because for mice, I think there should be data.
I don't know. I couldn't find.
Because for
I couldn't find anything.
For mice, actually, we can use a single age-related atlas that Eugene used in his paper because we have single cell data there. So
yeah,
for expression, yes. For expression, yes. I haven't looked at expression. It's a good point. I need to look.
So that's actually related to a question. So you only looked at cell number, right?
Yes, the frequency.
So yes, how precise is that? Because in the tabula myriacenis, I know they try to look at the frequency changes with age, but they don't even capture all cells, right? So they miss like a given number of cell types in a tissue, right?
So it depends. It depends on how you look. When you look with the normal cell counter, like a CBC machine, a normal blood analyzer, you miss a lot of cells, and it's not very accurate because these are very simplistic machines designed to give you some kind of clinical diagnostic very, very quickly. When you do it this way with flow, it's actually quite accurate.
Okay.
It's actually quite accurate as long as it's done correctly. And in this specific study, it was done correctly. A lot of the studies you will see it's not done correctly, but this one specifically, person did it was very, very good.
Yeah, so that was maybe using all data might be tricky, right? Because I'm not sure like people took care of that, right?
Yes, definitely. So, okay, so let me get back to this. The thing is, this is probably an overtrained model, right? It's very simplistic, but it's a proof of concept that I was trying to do.
How large was the test set?
In this case, for the neural network, it's 70% and 30%. So it's 120 and 40 for the validation. It's not, it was just, I was just playing with the neural net to see if it can even give any result. Okay, so now the more interesting thing. The reason this data is interesting, it doesn't just compare old and young, right? We just don't just do, okay, something goes down or goes up with age. We can look at the patterns of change. And here you can see different populations and how they change with it's a very simple OS regression, S-spline regression. But the thing is, we saw some interesting patterns so that we could look at. And especially you can look at the B cells and see that it's not just a linear reduction. It kind of seems like a logistic reduction of B cells with age. So we found that interesting, and I looked a bit deeper at the patterns. The first pattern I saw, by the way, is when I looked at variance or coefficient of variation as a measurement of variance. We see that variability increases with age, and it especially seems to spike around 2 years of age. So that's interesting. It indicates kind of a loss of regulation with age of population frequency. The second thing I did, I tried multiple regressions and compared them, with simple Bayesian statistics that the software does for me on its own. And I looked at what's the best regression to describe the pattern of change. And for some of them, I can define an inflection point, right? This is a mathematical inflection point of the regression, obviously, not of the data itself. The interesting thing is when I try and measure these inflection points. Another way to measure an point of change, which is not really an inflection point, but it's a point of change. You can do a change point analysis, kind of a multiple linear regression analysis, and find the breakpoint. You can also do this with a Heaviside function, but it doesn't matter. It's basically a regression where you find the breakpoint or the point where things seem to change. And when I did this for all of the parameters and played with the bit, I found something interesting. A lot of these change points or inflection points, they kind of converge around 15 months of age. So they kind of converge around the middle age. And when we look at that in terms of where it is on the survival curve of rats, it happens kind of here right before the drop in survival starts. And I think this is important. I think what this means is there's a point of change in hematopoiesis where population frequencies in the blood change dramatically, and that happens before the drop in survival starts during aging. And I think that is kind of highlighting a point of intervention where if we want to prevent the downstream effects of changes in hematopoiesis, we need to intervene before that. So we should at least try. And I'm trying now. I'm getting some rats that are about 12 months old and putting them on caloric restriction to see if it even does anything to these population frequencies. But this is, I think, the most interesting thing we found from this data.
But for caloric restriction intervention, it will be hard to compare with something, because if you do it at a different time point, the length of the experiment will differ, unless you do caloric restriction for a certain amount of time. But even then, the different stage will bias your results.
Yes, I'm to be honest, I'm doing it mostly as a sort of a proof of concept. The good scenario is that it works and it's proof of concept that I can delay some of these changes.
Yeah.
With just an intervention, caloric restriction, just as an intervention that's easy to do. And if it fails, I can say that changes in hematopoiesis are upstream to the metabolic pathway. But yeah,
So
you're right.
Regarding T cells, I'm just thinking about thymus, right? Because thymus involution, you also have this decrease, and so T cells are produced by the thymus, right? So I don't know if that could correlate these slopes.
Yeah, T cells.
So what is T cells?
T cell is the blue on the top. So we're kind of seeing a drop in overall T cell numbers, and then it remains pretty much constant for a while, and then it drops again around 2 years of age, a bit before.
Do we have any thymus involution data for this time period for rats?
Again?
Did anybody measure thymus degradation on rats?
I don't know. It's a good question.
But I'm not sure actually, even if the thymus is decreasing, number of T cell might stay stable because you have clonal expansion, but you have less diversity, right? So yeah, I don't know.
Yeah, so generally speaking, you have that, from what I know, right? You have thymus convolution that happens very, very early, starts very early. Then you're left with some very, very small thymus. But you're still seeing we're still seeing a lot of T cell generation, right? Even in the bone marrow. They all go through the thymus eventually, but we're still seeing a lot of generation of T cells. I don't know much more than that.
Okay, I don't know.
Yeah, just to clarify something, all these curves are from the same animals, right?
It's not a longitudinal study. It's a cross study.
Yeah, so no, but it's cross study on the same population. I mean,
yes,
the T cell curve and the blue and the purple is from the same
from the same animals, and because it's frequencies, they're not independent measurements.
Mm-hmm.
That's a big downside of the data set. Yes. I hope the reviewers are not as keen as you. All right, so the last thing we looked at is the DNA methylation. Those of you that don't know, DNA methylation is an epigenetic regulation where you get methylation of bases on the DNA, mostly of C. And you can easily look at DNA methylation when you do bisulfite sequencing. You just do a bisulfite reaction on the DNA that you take, and that converts unmethylated C into U. The methylated ones stay protected as C, and then when you sequence, you can say which sites were methylated and which ones were not. And we did a lot of methylation analysis that I don't have the time to go into, but the interesting thing, I think, I did the same analysis of change and looked for genes or promoters where I see some kind of inflection over breakpoint in their change. And I saw that the ones that have a breakpoint early in life, before 15 months of age, they have something to do with regulation of hematopoiesis, ApoE and RUNX1. That I think is very, very important. At least it's very important for us because we're looking at peripheral terminally differentiated cells in the blood, right? But we're seeing altered DNA methylation of genes that regulate hematopoiesis, even early on the hematopoietic stem cell stage. So that to us indicates that a lot of the population frequency differences that we see with age, they result from changes in the stem cells. And it's not just terminal regulation. We think it starts in the bone. So that's the basics of it. There's a lot more, but it will take a long time to present. But I just wanted to give this a preview. And that's it. Thank you to everyone for listening. I know this was long and the last talk of the day. And thank you for the comments. There were some very, very good ones.
Thank you for the talk, Hagai. I don't know if there is another comment or question.
One small comment.
Yeah.
Okay. Just this work once more showed the importance to look into how we call this age, middle age. Okay. It is critical, but it didn't receive the attention it should receive. And I have not one example that it is really important. One of them Hagai knows very well because he was the first author in this paper. Also, it was a paper on wound healing. And again, the first changes were found exactly in the middle age. Now, old age is actually the result, but each crucial, dramatic things actually happens in the middle age. Okay, nice.
Okay.
Publish this work.
Working
Why
on
are
it.
you waiting? I don't know.
I'm writing it now.
Writing. Okay, writing is good.